So now that we've considered the external structures of prokaryotes, as well as components of their cell membrane, let's take a look at their internal structures. We'll begin by looking at the cell's cytoplasm. The cytoplasm in general is a gelatinous solution. It's about 70 to 80% water, and it's the site of very many of the biochemical and synthetic activities that occur inside the cell. Within the cytosol, or the liquid component, we have many other components that contribute to the cytoplasm. They have things like cell masses, like the chromatin body, the ribosomes, the granules, and actin and tubulin strands that act as the cytoskeleton in some of the bacteria that have those components. Taking a closer look at these cell masses, we'll first consider the bacterial chromosomes. Bacterial chromosomes, as you're familiar, are single circular strands of DNA. They're long strands of regular double-stranded DNA. It's just one single molecule as opposed to paired molecules that we see in the human condition. They have one single circular chromosome, whereas we have 23 different pairs of chromosomes. This DNA is coiled around protein molecules in order to fit into their nucleoid compartment. This nucleoid region is an aggregated, dense area in the cell where we see the DNA arranged. The DNA contains genes along its length, and these genes code for information that's required for growth and maintenance of the cell. In addition to the mandatory prokaryotic chromosome, many bacteria contain other pieces of DNA beyond this essential chromosome that are not essential to growth and metabolism, but may confer some protective traits like antibacterial resistance, or they may produce toxins that protect the cell from things in the environment. Again, these are single molecules of DNA, and all DNA is double-stranded. They're very small fragments, and they exist as separate circular units. On occasion, they can be integrated into the bacterial chromosome and then duplicated during conjugation so they can be passed on to the progeny of the individual cell. Next, we'll look at ribosomes. Ribosomes are made of RNA and protein. They show up in the cytoplasm as tiny specks or in chains called polysomes. As one piece of RNA is translated by multiple ribosomes, they appear in a strand-like structure as they translate the RNA strand into proteins. Ribosomes are made up of about 60% ribosomal RNA and about 40% proteins. They have two different subunits, a large subunit and a smaller subunit here. Now, we characterize ribosomes by S units, or Svedberg units. These units are used to rate the molecular sizes of the ribosomes. When we spin the ribosomes down in a centrifuge, they separate by molecular weight. Those that are most dense sediment or fall to the bottom faster. They have an higher S value. Prokaryotic ribosomes are 70S. That is, both subunits together weigh 70 Svedberg units, whereas eukaryotic ribosomes are 80S. The importance of the distinction between 70 and 80S ribosomes will become more evident as we move through the content of this semester. In addition to the other organelles, there are inclusion bodies inside prokaryotic cells. These inclusions can be either membrane-bound inclusions or they can be granules. The membrane-bound inclusions are usually for storage of nutrients. We'll have things like volutin granules, which are for storage of inorganic phosphates that can be used for manufacture of ATP. Or we might have inclusions for the storage of carbohydrates, such as glycogen, or other carbons in the form of polyhydroxybutyrate granules. These storehouses of nutrients can be used in times when the environmental sources of the nutrients are depleted. So the bacteria essentially store nutrients in inclusions for use later. These inclusions can be in varying sizes and number and content. Some of them have a single layer of membrane, and other granule inclusions have no membrane at all. You can see the polyhydroxybutyrate ones in pink up here. Sometimes these inclusions might be gas vesicles that are used for flotations, or a really interesting one here is magnetosomes. The magnetosomes we can see here as little black dots along the edge of this bacterium are magnetic iron crystals. They're not involved in the nutrition at all. They're more involved in cell orientation, and they act to pull a cell towards polar or gravitational fields, so they aid in the movement. Some other considerations of granules would be these non-membrane bound granules. They consist of crystals of inorganic compounds. For example, there are sulfur granules that we see in photosynthetic bacteria. Polyphosphate granules of the Corynebacterium and Mycobacterium are called metachromatic granules because they stain a contrasting color when we stain them with methylene blue. Finally, let's consider the cytoskeleton. We used to think that bacterial cells held their shape exclusively because of the structure of the peptidoglycan cell wall that we've already examined. And this is true of many bacteria, for example, the cockeye-shaped or spherical-shaped bacteria. But others produce actin and tubulin molecules. These are found in spiral and rod-shaped bacteria and aid in them holding their form. They're arranged in helical ribbons around the edge of the cell, just under the membrane. And they may be involved in how peptidoglycan is secreted into the cell wall area. Let's take a quick concept check. Where would you find the genetic material of a prokaryotic cell? In the nucleus, in the nucleolus, in a nucleocapsid, or in the nucleoid? Well, the nucleocapsid didn't really even come up, so we'll get rid of that one. And the nucleus is found in eukaryotic cells. By definition, prokaryotic cells don't have a nucleus. The nucleolus, although we haven't covered it yet, is an area where the DNA is located inside the nucleus. So that cannot be included. So that leaves us with the nucleoid. DNA is indeed found in the nucleoid region in prokaryotic cells. So there's certainly a lot of evidence that bacteria are able to protect themselves against adverse habitats. But of all the microbial structures, nothing really compares to the endospore. It's a unique structure that we see in some prokaryotes. Now, a note about the word spore before we begin. It has more than one use in microbiology. It's defined as being a tiny compact cell produced by either vegetative or reproductive structures of microorganisms. In this case, the endospore, or spores produced from inside the cell, generally function in survival, not reproduction. Now, there's an occasional exception. We'll take a brief look at these later. The endospore structure helps some bacteria survive extremely adverse habitats. And this spore formation, or sporulation, is induced by environmental conditions. Say, depletion of essential nutrients like carbon and nitrogen. So these dormant bodies are produced by Bacillus, Clostridium, and Sporosarcina. The endospore-forming bacteria have two life cycle phases. Phase 1 is the vegetative cycle. That's where they're living a normal metabolic life. They're metabolically active and they're growing. However, when the environment becomes too harsh and major nutrients are missing, they can be induced to undergo sporulation and enter phase 2, or the endospore phase of life. Let's take a quick look at how that happens.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolenic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell, and the mature spore is released.
Formation of the spore from the vegetative state takes about 6 to 8 hours. Once the free spore is released and the sporangium is lost, it's in the most hardiest of all life forms. It can withstand extremes in heat, drying, freezing, radiation, and chemicals. This heat resistance is possibly due to the high content of calcium and dipicolinic acid. Heat inactivates proteins and DNA, but this process requires water. The role of the calcium and the dipicolinic acid is to remove water from the endospore. Without this water, it's more resistant to heat as well as drying. Some viable endospores have been found that are more than 250 million years old. So essentially, this form has almost infinite possibility for survival. Once favorable conditions arise again, the endospore will revitalize or germinate. Germination requires not only water, but a specific germination agent. This agent might be something as simple as an amino acid or an inorganic salt. This combination stimulates hydrolytic enzymes in the endospore's membrane. These hydrolytic enzymes digest the cortex and allows the core to rehydrate, take up nutrients, and grow out of the endospore coat, as you can see in these steps here. This process only takes about an hour and a half, as opposed to the 6 to 8 it took to form the spore. Now, this process is not considered reproductive because most bacteria only produce one endospore, and that one endospore will only germinate to form one vegetative cell. There are some exceptions to this rule, however. For example, Metabacterium polyspora, which is found in the intestines of guinea pigs, can form up to 9 different spores. So in this sense, it could be considered reproductive. But in general, endospore formation is not a reproductive function. Now, both gram-positive and gram-negative bacteria can form spores, although all the medically relevant ones seem to be gram-positive. Most of them are relatively harmless. However, some are not. We'll cover many of these non-harmless examples as we look at the various infectious diseases of different systems and produce our wikis, but here are a few examples. Bacillus anthracis, you may be familiar with. We had a little issue with that not so many years ago. The presence of this endospore is very persistent, and it makes an ideal candidate for bioterrorism because it can exist for a long time in this spore state. And only when it's presented with the ideal situations will it germinate again. For example, when it contacts the membranes of respiratory passages. Clostridium tetani that forms tetanus, and Clostridium perfringens that causes gas gangrene are other examples. When these spores are embedded in dead tissue, they germinate and grow. They release potent toxins, which cause all sorts of issues. Another profound one you may have heard of is Clostridium botulinum. It causes botulism, the deadly form of food poisoning. When food is not canned properly, these bacteria can really wreak havoc. Endospores are constant intruders in our environment because they inhabit the soil and they're present in most dust. They resist ordinary cleaning methods. So, for example, in canning, you can't just boil the products you wish to can because the spores can exist far beyond 100 degrees centigrade. So they have to be boiled in pressurized steam at 120 degrees centigrade for up to 30 minutes. This prevents them from causing food spoilage and poisoning that we might see in old canned food that have caused botulism commonly in our society.